This is where we left off on the last one, and I was getting pretty excited about telling you about this duplication of the olfactory genes that I kind of messed up and anyways, so sorry. But the real exciting thing is this particular gene sequence has been duplicated so many times, and it's in the humans also. But remember, it's redundant, so the duplication doesn't cause a huge problem. But the thing of it is it causes a benefit that's very subtle. We find that in species that tend to use their sense of smell, like mice and other species that maybe hunt at night or use smell for many things, not only just finding food, but also realizing if there's danger. This gene sequence has been duplicated many more times and in humans. And we do see, and what this graphic is showing you is this increase in olfactory size has actually corresponded more or less with the increase in body size of species as they've gotten larger. But certainly we find that those species that use their nose, which we observe them to use their nose, it's been duplicated far more in their species. Now, rapid reproduction can cause increase in a chance of mutation. We've seen this in other chapters. So mutation rates are often higher in prokaryotes, or higher in viruses, and lower in eukaryotes and prokaryotes. Sexual reproduction is one really great way to shuffle alleles into new combinations. Remember, with sexual reproduction in meiosis, you have that crossing over that you can have allele locations actually change or exchange allele locations on the same chromosome. So you have this new combinations that can take place because of the making of the gametes. We find that those organisms that produce sexually, it's more expensive energy-wise, so it's much more complicated, expensive, takes a more sophisticated species, but their payoff is the fact that you get this great variety of genetic recombinations. This is found to be more important than mutations in eukaryotes, in species that sexually reproduce, than mutations. And in testing all of this can be tested as to whether a population is evolving or not, based on one gentleman's equation, Hardy-Weinberg. And so what you have here are the population. If a population is defined as a group capable of interbreeding and producing viable offspring, so fertile offspring. That fertility is both on the male and the female side, and each variation can unfortunately produce a problem with each sex. A gene pool consists of all the alleles for that locus in a particular population. I believe they pronounce that word loci, but I always just say locus, sorry. The locus is fixed, and actually the difference between loci is plural, locus is singular, is fixed if all the individuals are homozygous for that allele. So it's fixed if all the individuals are the same for that allele. And so you have this population defined as a group of individuals that are interbreeding. if they're all interbreeding and they have the same allele, then there is not going to be any variety that is going to happen unless there's a mutation, unless that's a harmless mutation. So that's what we're talking about here. And then further delving into this Hardy-Weinberg principle, Hardy-Weinberg came up with an equation that could calculate whether the frequency of an allele, what it is, and calculating it. And the equation is here. I'm not going to ask you to actually calculate an equation, but I do want you to know what Hardy-Weinberg is, and you do want to know what the equation is. So what the equation is, it equals 1. That means it equals the sum total of the whole population. So and it will be expressed in percentage, where you have sexual reproduction, so you have 2 alleles. You're going to have a dominant and a recessive. That's where the big A, little a comes in. The frequency of the dominant allele is represented by P. The frequency of the recessive allele is represented by Q. And so the frequency of a genotype, homozygous dominant, is P squared. Frequency of genotype homozygous recessive is Q squared. And the frequency of the heterozygous dominant recessive is 2 times PQ. And so again, that equals 1, and you come up with the equation that you have above, P squared plus 2 times PQ plus Q squared equal 1. And so with that, if you know what P and Q are, you can calculate the percentage of the unknown. So you have to know what everything is but one of the unknowns. But using this equation, it can be used to calculate the percentage of that particular allele. And the principle, here's the gentleman's name, Hardy-Weinberg, describes a population that's not evolving. And if a population isn't evolving, since the environment constantly is changing, if the population isn't evolving, then eventually it's going to be unfit for its population. So a non-evolving population tends to be tends to be a negative. We need to be continually adapting to our environment, and there is certain criteria for the Hardy-Weinberg principle that is concluded that is then calculated that a population is evolving or is not. And the principle states that the frequency of alleles and genotypes in a population remain constant from generation to generation. That populations where gametes contribute to the next generation randomly, frequencies don't change. Now, this isn't a new concept. We learned this with Mendelian inheritance, that you have this randomness, you have this 50-50 chance, this 50% chance that a particular allele will show up. And so that's what the idea here is. If you start out with a certain percentage, for example, in this image, you have 80% red balls and 2% white balls, that the 50-50 chance is that you're still going to have 80-20 in the percentage between the egg and the sperm. That's Mendelian genetics. So the gene pool doesn't change according to Mendelian genetics, so something's going to come along to actually alter the gene pool, and we'll talk about that. But Hardy-Weinberg equilibrium describes a gene pool that is stagnant, that is not changing, and that's what the equation is for, is to see if a gene pool is changing or not. And you have calculations here. You have an excellent example in your textbook about a protein that either PKU and the frequency in the United States, fortunately, because the protein PKU, and if somebody inherits this disorder, they're not able to metabolize a certain protein, and in the past it's been very deadly. They do have some treatments for it now, but the percentage of people carrying that allele is about 2%, about 0.02%, 2%, sorry. And then in terms of a little bit more about Hardy-Weinberg, certain conditions have to be met in order for a population to not be evolving. So in Hardy-Weinberg equilibrium, 5 conditions. There have to be no mutations, and we already talked about that in the fact that generally there are mutations. There has to be random mating. Well, random mating generally takes place. No natural selection, and so the chance of that is going to be very, very slim, that there's going to be no natural selection. Extremely large population, that can be very likely. And no gene flow, meaning nobody is trying to go into a new environment because there's food there, food sources there, or maybe there isn't as many dangers there. So all of these conditions have to be met in order for that population to be non-evolving. This happens very rarely in nature, that all of these are met. The only time that it really is we see it in very small populations of, say, large cats, because other stressful conditions, for example, maybe their territory has been reduced, or maybe their populations have been artificially reduced. So we do see some populations of large mammals that are in Hardy-Weinberg equilibrium, and things have been trying to be done about it. And if you haven't seen all the Amoeba Sisters cartoons for the various concepts with regard to biology, it's good to check them out, because they do a really good job of explaining things in a very funny ways. 3 main factors that alter allele frequencies are and bring about most of the evolutionary change are natural selection, which we've talked about quite a bit, genetic drift, and genetic flow. And so the success with regard to reproduction will then pass those alleles on to the next generation. You have a couple of examples of this. You have the moths alighting on the tree, and the white moth is going to stick out, so it's less likely that that white moth is going to pass its genetic information on, because it's going to get eaten. And then unfortunately, if the environment changes and now the environment favors the white moth, it's the dark moth that's going to not pass on its genetic information. Another example is DDT resistance. We found that it was used widely in agriculture, and it created these DDT resistant problems. Whatever it was trying to kill, found that you had killed off all of the organisms that had no resistance, and all you had left were the ones that are kind of like the bacteria, the antibiotics. Genetic drift describes how allele frequencies fluctuate unpredictably from one generation to another. And it does reduce genetic variation through losses of allele. And I thought this was kind of graphically maybe a little violent, but anyway, so you have a 5-to-5 ratio on the far left from blue to red. Unfortunately, something comes along and knocks a good portion of the blue ones off, and then the reproductive ratio, again, the Mendelian genetics 50-50, but if there's only one blue one to reproduce, then you're still going to have just 2, but 2-to-10 ratio then. So genetic drift can alter the allele frequency that way. Then you also have a couple of other things. We'll get started on this one. Founder effect. You see founder effect quite a bit on the Galapagos, where you have certain species have become isolated from a larger population, and then that particular species will populate that area. You also have that smaller founder population can be different than those in the larger in terms of evolution. We'll pick this up on the next one.